Hello and welcome to Exam 3, Essay 3. In this video, I will be going over the components of blood and plasma, as well as RBC or red blood cell breakdown. And then lastly, I will be covering hemostasis, specifically how platelets are activated and how coagulation or blood clotting occurs. So to begin, I'll start off with blood. So imagine this is a tube, and this tube is filled with blood that's just been centrifuged. So I'm gonna go ahead and color code this. So the red is gonna represent our blood plasma, and the plasma is gonna make up about the majority of the blood, and so it's gonna be about 60% of it. The plasma is also gonna have its own separate components, namely we've got albumin, which is involved in BCOP, which I've explained in a different video. We've got globulins. And basically what those are is they are just another word for antibodies, because we have a lot of antibodies in our blood, specifically from white blood cells. And lastly, we have fibrinogens, which are involved in blood clotting. So we just call them clotting factors. And then the next component of blood, I will label in black. These are called forming elements. And by that, we mean the cells themselves. So we've got red blood cells, which we abbreviate as RBCs. We also have white blood cells, and we've got so many different types of white blood cells, but I won't get into that because that would take a really long time. And then we also have platelets. And that's pretty much it for the components of blood and plasma. When plasma does not have all these proteins in it, we call it serum, by the way. Then the next thing I want to go over is how red blood cells are breaking down. So if we imagine, actually let me use red for this. If we imagine a red blood cell, they're going to be made in the liver, the bone marrow, and the spleen. And before they are made, they are going to be known as reticulocytes, which is basically just a premature red blood cell. Red blood cells have 8 subunits. 4 of them are globulins, and 4 are what's called heme. And heme is very important because it's going to contain iron, which gives it its red color, which is also going to help in oxygen binding when we breathe in and get oxygen into our blood. So now, when we break down our red blood cells, they're going to be broken down by macrophages, which is a white blood cell that basically just eats stuff. So it's going to be broken down. All of those components, globulins and heme, are going to be separated into its parts. So we've got heme and we've got globulins. The globulin remains are going to just re-enter the bloodstream and build itself again, whereas heme is going to go through a very long different process that's going to travel to our intestine, to our liver, to our intestines, and to pretty much our entire digestive system. So to start, heme is going to be converted into a component called biliverdin. And so after that happens, it's going to become bilirubin as it is transported from the blood into the liver. But this bilirubin is going to be unconjugated until it hits the liver. So now we're in the liver. When it gets into the liver, it's going to be converted into conjugated bilirubin. And how that happens is through something called glucuronic acid. Once it gets conjugated in the liver, it is going to be secreted into our bile, which is what the liver is going to produce. And then that is going to get secreted into our intestines. And that is where we lose our glucuronic acid. And once that happens, it's going to form something called urobilinogen. Now, once it hits this stage, it's going to go 3 different pathways. The first pathway is that urobilinogen can re-enter this whole system and go back to the liver and basically recycle itself. The second pathway is it goes to the kidneys, where it's going to form the yellow urobilin. The other pathway is it stays in the intestine and becomes stercobilin, which is going to have a brown color. And then finally, I will go over platelets and hemostasis. So I'll go ahead and erase this. So if we define hemostasis, that's essentially the cessation of blood, as according to the textbook, or basically stopping blood flow when we start bleeding. There's 3 different ways that happens. One is vasoconstriction, which we all know that's just the constriction of blood vessels, so it will cause less blood to flow. The second one is platelet aggregation, which I will explain with this picture here. Imagine this is a blood vessel, and imagine these here are epithelial cells that are lining that blood vessel. So these are epithelial cells or epithelial tissue. And then over here up top, this is called a basement membrane, which is composed of collagen. So what happens is, say we get a wound or a break in our skin, which is going to cause this to open. So we have all these platelets, and they're going to have a bunch of different receptors that play roles. So the first receptor I want to go over is something called VWF. This receptor is going to cause the platelet to bind to the collagen. So this is going to bind to the collagen, and let me draw it over here. So our platelet's going to bind, and it's going to get activated. So the whole shape is going to change and kind of look more like this. And then once that happens, our platelets, which contain dense granules, are going to release ADP out in the open. And once ADP is released, we're going to have more platelets that have ADP receptors. These are going to grab ADP, and then they're going to start attaching to the platelet that was already attached to the collagen. And then another type of protein that we're going to see is something called TX2A. And that's going to be a receptor for something called thrombin. And once this binds, that's also going to come over and bind to our activated platelet. And then the next thing you know, we have all these activated platelets forming a platelet plug onto our vessels. And so that's our platelet aggregation. The last thing I want to go over is coagulation, which is going to involve a cascade series of how the clotting of blood will happen. So there are 3 different pathways. We've got extrinsic, intrinsic, and common. So I'll introduce our intrinsic one first. So let's say we have our collagen, and we've got our epithelial tissue. So basically with this cascade, there's going to be a ton of proteins that we're going to refer to as factors that are going to be converted until the clotting system is formed. So our intrinsic is going to have factor 12 become factor 12A, and that's going to happen as it travels to collagen and then becomes factor 12A. And then that's going to cascade to factor 11, which is going to become factor 11A. And then that's going to cascade to factor 9, factor 9A. And then I'm just going to go ahead and write this over here, not to confuse you and to have more room. And then that's going to cascade into factor 10 becoming factor 10A. And at that stage, we're going to hit what's called the common pathway. And so the common pathway, I'll describe in just a moment after I go over extrinsic, but intrinsic, we call it this way because pretend this is happening inside the vessel. I drew it outside, but it's called intrinsic because it is happening inside the blood vessel. The extrinsic pathway, however, does not happen fully inside the blood vessel. So I will go ahead and draw the extrinsic. So for extrinsic, we have a protein up here called tissue factor, or TF, which is also known as factor 3. That is going to change factor 7 to factor 7A. And then that alone is going to jump into our common pathway. So our common pathway, we're going to start from factor 10A, and then that's going to become factor 2 is going to become factor 2A, which we call thrombin, and then factor 2A is going to be factor 1, which is fibrinogen. I can't really write that because I don't have enough room, but fibrinogen, which is going to become factor 1A, which is going to become fibrin. And this is the protein that is involved in our blood clotting. And then the last thing I want to go over is some of the tests that are involved in intrinsic, which I'll label here, and our extrinsic factors or pathways. So the tests that are involved are, for intrinsic, we have heparin, which is a protein that will inhibit this. And the way we can test the blood clotting efficiency for intrinsic is something called PTT. For extrinsic, it is going to be PT, and the things that will inhibit it are things that inhibit vitamin K, because that's what's going to cause our extrinsic pathway to occur, and that's going to be called condin and warfarin. And that's what's going to inhibit vitamin K, to inhibit our extrinsic factor. And the reason we have this is so that our blood does not overly clot, because when our blood clots excessively, that can be very lethal and could eventually cause death if the blood clot gets to like our brain or our lungs or something vital that won't allow enough blood to pump through. And that is it for my discussion on exam 3, essay 3.